All right, so today what we're going to do is we're going to cover, so our first day, our first two sessions, we were looking at Partec. And what we did was we used Partec to analyze a public data set having to do with stem cells. Not only stem cells, but the difference between pluripotent stem cells, which are from adults, and embryonic stem cells, which are from embryos, obviously. And we also looked at the differences between when they're just at their normal state and when they're induced for differentiation, okay? So we used Partec. We found that data set. We got all the sample information. We merged those two files. We created a file that had numbers that were related to the levels of expression in those genes. And in fact, that was 55,000, or no, it was about 36,000 different genes. We visualized this information. We found out that part of the study was probably done at a different time, and so that we weren't able to analyze that together. So we eliminated the outlying study, and so we were left with, I believe, 18 samples, or was it 9? I can't remember. I think maybe it's 9. It is 9, isn't it? No, wait, 18. 18 samples. Just containing pluripotent stem cells and embryonic stem cells at day 0 and day 5 when they're differentiated. We used the ANOVA to find out significant genes that were different between time 0 between the two different cell types, day 5 when they were differentiated between the two cell types, and we also used it to find genes that change, their expression changes due to both factors. So both day and cell type. That was a two-way ANOVA. When we did that, we found lots and lots of genes that were different. So to kind of normalize everything, we took the top 1,000 that were significant, and we did a Venn diagram. Let me put that up real quick. There it is, Venn diagram. So we selected our day and cell type, also those that were different at 0 and day 5. So from these gene lists, what we were able to do is make subsets of gene lists. The one I'm going to talk about today is this group, this 25 genes that were common to all these conditions. So again, in these 25 genes, they showed a difference in expression levels at day 0, day 5, and they were affected by both cell type and by the differentiation state. We're also going to talk about, is that D? Yep. This list too, this 1,000 genes that have both cell type and differentiation effects. Has everybody loaded in your gene list? Oh my gosh. If this was my high school class, half of them were like, we were supposed to do that? This is awesome. Yeah. It's nice teaching grad students for a while, for sure. Okay. So you should have those gene lists in your IPA. Everybody has their IPA opened up, right? I'm going to tell you right now that this is where you make your money in bioinformatics. This is my favorite part. Yeah, it is my favorite part. I was going to say, well, maybe not. No, it totally is. That this is where you incorporate the biology, that these aren't numbers that we're just looking at. These have histories. These are genes. These represent genes, and these genes have buddies, and they have interacting partners, and they have enemies, and it's all in the scientific literature. So what IPA does, and the reason why we're downloading our gene list into IPA is IPA probably has the deepest, most well-curated database in the world. I've told you the legend that there's a bunch of postdocs chained to their computers on an island outside of Silicon Valley, and they're just going through the scientific literature trying to characterize all these correlations. The one thing that sets Ingenuity apart versus some of the freeware solutions is that it's human curated. That means this isn't some kind of algorithm that puts all this stuff together. And most of the freeware stuff, the bioinformatic packages are all about freeware, or about text mining. This actually, you have a human individual going through the data, going to the papers and saying, yes, this gene interacts with this gene in this paper. All that information is tied. And so when we put our gene lists in, basically we send those gene lists to the supercomputer in Silicon Valley where Ingenuity is. They take all their information they know about those genes, basically add that information to your gene list, and then send you back that information and give you this platform to visualize that. Okay? Again, I think that this is where the future of bioinformatics is going. That, anybody can polish numbers. Like me creating a gene list, doing a bunch of t-tests, like that's pretty standard stuff, This is where you make your money is using that information. This is kind of the art form of bioinformatics, is how do I take that biology and make a picture or make a structure or a pathway that's actually useful. And that's kind of why I came to you bioengineering students, is because I love the fact that when you get data, you want to do something with it. You want to make it work, like build something. Most people, I think a lot of computer science people who get in bioinformatics, they spend a lot of time and energy developing a specific tool to do their work, right? They build their hammer. And they spend so much time building their hammer, they're going to use their hammer on anything. Screws, nails, darts, whatever. They're going to hit everything with their hammer. What I like about this software, IPA, is you can use it for anything, It's a very good tool, and it relies, the more biological information that you have, that you bring to the table, the better IPA will perform for you. Okay, well, I've been talking this up for about 10 minutes, so let's see what the heck it does, right? Let's drive this car.